So welcome back from spring break. We will essentially pick up right where we left off. So we'll head into the citric acid cycle. We're going to spread the citric acid cycle up over 2 lecture sets. And so this is the first of those 2 sets. We're covering the first half of chapter 19 from the textbook here. And essentially we're going to start off reviewing glycolysis. I think it's been a bit of a while perhaps since some of you thought about this material. So I want to make sure we're all starting on the same page. And of course, that's the page of 2 pyruvates made from a single glucose molecule. From there, we'll take a big picture view of the citric acid cycle and talk about its overall role in metabolism in general. And then we'll have 2 other chunks after this one, B and C, where we'll start off with a brief but thorough overview of the citric acid itself, and then we will get into the citric acid cycle with more detail. We'll start off talking about how we convert pyruvate into a molecule called acetyl-CoA. It's actually acetyl that we feed into the citric acid cycle, so we're not even in the citric acid cycle until we've made acetyl-CoA. And from there, in the very last lecture chunk, we'll talk about the first half of the citric acid cycle and leave the second half of the cycle as well as its regulation for the next lecture in this series. So again, since spring break has passed and we kind of left off on a cliffhanger in many ways, let's talk about glycolysis once more so that we get it fresh in everybody's memory. We're going to be picking up right from where we left off here, so let's make sure that we're comfortable with glycolysis. Yeah, how did we get from 1 molecule of consumed glucose to 2 molecules of pyruvate? Once we review that and we have that pyruvate, remember we have to turn that pyruvate into acetyl-CoA. And then it's that acetyl group and that acetyl group alone that we feed into the citric acid cycle. You may recognize CoA as our activating coenzyme A, and indeed that's what it's doing here. Acetyl is a very happy and content little molecule, 2 carbons big, and the only way we can convince acetyl to get into the citric acid cycle is to first pair it up with the worst blind date ever, which is CoA. And then acetyl is so anxious to get away from CoA that it will engage in the chemistry of the citric acid cycle. As I mentioned, as we'll see here and in the next lecture as well, so actually spanned out across all of these lecture chunks, the citric acid cycle is far more than just a player in glucose metabolism. It is in many, many ways the central hub of all eukaryotic metabolism. We'll emphasize that point in many different ways. But almost every metabolic process either feeds into or is fed by the citric acid cycle. And again, we'll explore that a little bit more in depth later on in these lectures. But for now, what I'd like you to think of the citric acid cycle as is a mechanism or a step in glucose metabolism, and more specifically, I'd like you to keep one thing in mind. The citric acid cycle is an electron harvesting process. The citric acid cycle exists in glucose metabolism to rip electrons from the carbons of glucose, now in pyruvate, and use those electrons for ATP synthesis. That's its primary role in glucose metabolism. It is one step getting from glucose to ATP production, and I want us to always keep that at the fore. The other roles that the citric acid cycle plays in metabolism are important, but no more important by any means than its role in harvesting electrons from glucose. So we've made this point many times already, starting with the redox lecture. But in metabolism and living cells in general, electrons are energy. Electrons are the physical manifestation of energy. When we move electrons with redox reactions, we are moving energy. And so when the citric acid cycle is stealing electrons from the carbon of glucose, it's actually stealing energy from those carbons. But again, let's start off where we're comfortable. Let's review glycolysis very briefly. We started off with stage 1 of glycolysis, where we consumed glucose, phosphorylated it to glucose 6-phosphate, isomerized that to fructose 6-phosphate, and then phosphorylated fructose 6-phosphate to fructose 1,6-bisphosphate. Steps 1 and steps 3 there consumed ATP, and we were at a 2 ATP deficit at that point. Fructose 1,6-bisphosphate was then cleaved into GAP, or glyceraldehyde 3-phosphate, and DHAP. And DHAP was isomerized into another molecule of GAP. So at the end of phase 1, or stage 1 of glycolysis, we are poised with 2 molecules of GAP from that single molecule of consumed glucose. Then, in stage 2, we take those 2 molecules of GAP, now everything we do in parallel, and we phosphorylate them to 1,3-bisphosphoglycerate, 1,3-BPG. We don't need ATP for this because we're using the energy of the redox reaction to power this reaction forward, and we use inorganic phosphate as our phosphate donor. Then we take one of those two phosphates from 1,3-BPG. Remember, this is a molecule with a high phosphate transfer potential. And we use it to phosphorylate ADP into ATP. We make two there because everything we've done in parallel, so we just broke even from an ATP perspective. But when 1,3-BPG loses its phosphate, it becomes 3-phosphoglycerate, or 3PG. We then engage in what we are perfectly content to call an isomerization reaction, actually a mutase reaction, where we move that phosphate group from carbon 3 to carbon 2, creating 2PG, and then via a dehydration reaction, we convert 2-phosphoglycerate into phosphoenol pyruvate. PEP, or phosphoenol pyruvate, also has a high phosphate transfer potential. It loses its last phosphate to ADP, making 2 molecules of ATP, our net gain of 2 molecules of ATP, in fact, and what we are left with is 2 pyruvates. So the end goal of glycolysis is to, in this 10-step process, create 2 molecules of pyruvate for every 1 molecule of glucose consumed. So, great. What have we accomplished? Well, we've taken a 6-carbon molecule and created 2 3-carbon molecules from it, but the big question becomes why? Why did we do this? And what do we do with the pyruvate we just produced? Well, the short answer is, at least as far as the glucose metabolism story goes, we are going to convert that pyruvate into acetyl-CoA so that we can feed acetyl into the citric acid cycle and rip electrons from it, stealing energy that was previously stored in glucose along the way. So the role of the citric acid cycle in metabolism is multifaceted. We've got its role in stealing electrons, which we will focus on, but it's also involved in catabolism and anabolism, breaking down other molecules to energy and using precursors of the citric acid cycle to build the molecules we need to survive. Now, that central role of the citric acid cycle, the energy producing role, is essentially an aerobic respiration step. So the aerobic respiration of glucose is used primarily in eukaryotic cells because it allows more energy to be consumed from glucose, to be stolen from glucose, than any other metabolic pathway that has currently evolved. The anaerobic mechanisms that we've discussed in the previous lecture, where we made lactate in muscle cells or ethanol in fermenting cells, can sustain life, but it can't create energy at the levels needed for multicellular life, for life such as us. So we need aerobic respiration in order to make enough ATP to survive. The only problem is that aerobic respiration requires high energy electrons. And the only real way we have to harvest high energy electrons is by using the citric acid cycle. So we need the citric acid cycle as an electron generator, or as I said before, an electron harvester. Essentially, by the end of this story that we tell, we will make between 30 and 32 ATPs for every single glucose molecule we consume. Think about that. We'll take a single glucose molecule and make over 30 ATPs from it. And we're going to do that largely with the energy contained in the electrons we are about to steal. Along the way, because we're stealing electrons, we are oxidizing those carbons of glucose. Fully oxidized carbon is carbon dioxide, no surprise there. And so the carbons that we exhale, in fact the carbons you are exhaling right now as you listen to me speak, are the fully oxidized carbons of glucose you have previously consumed. We talked about water as being the wastebasket of the depleted electrons that we stole. Protons go with them. And so with each breath you exhale, you are exhaling both the spent carbons and spent electrons from consumed glucose. Pretty amazing. As I mentioned, the citric acid cycle is also involved in breaking down other consumed molecules to release energy. Fats and lipids are also eventually fed into the citric acid cycle. That's all catabolic. And it also plays a role in making molecules. So we will steal precursors from the citric acid cycle and use them to build biomolecules that we need to survive. And so the citric acid cycle plays a very big role in anabolism as well. Again, this idea of it being a central hub of metabolism. So if we come back to this figure, which we've talked about before, we are kind of following the trajectory of this single pathway or this single branch through here. We won't be talking about anaerobic respiration at all anymore. And so we have left our story off at the production of pyruvate from glucose, and now we're ready to stroll into the citric acid cycle. Now this is the same figure essentially, just a little bit more detailed, and to orient us here, we are on this first track. We consumed glucose, and through the process of glycolysis, we convert that glucose into pyruvate. Pyruvate needs to be converted into acetyl-CoA, and acetyl-CoA can be fed into the citric acid cycle. And you'll notice what's coming off of the citric acid cycle along the way are electrons that will eventually be fed into an electron transport chain, which will allow us to make the ATP that we're after all along. Once those electrons are depleted and done, they are added to oxygen. And when added to oxygen, those electrons and the protons that follow them will create water. Now I'm showing you this to point out one thing though. Look what else we can turn into acetyl-CoA. We can metabolize fats into acetyl-CoA, and we can metabolize amino acids into acetyl-CoA. Once we make acetyl-CoA, we can feed that into the citric acid cycle and generate ATP from it. So the citric acid cycle, and I guess by definition we can say acetyl-CoA itself, serves as the central catabolic hub. Anything we can eat, we can convert into acetyl-CoA and get electrons from it. It's just that it's most efficient with glucose. So let's continue on from pyruvate. Everything we've discussed so far with regards to glucose metabolism has been cytosolic. It's occurred in the cytoplasm. That includes importing glucose from the bloodstream. Glycolysis itself is all cytoplasmic. Everything we've talked about. The citric acid cycle, and pretty much everything else we will discuss from that point moving forward, however, is mitochondrial. So we'll talk about the mitochondria just a little bit to get our bearings there. As I'm sure you've all heard before, either in high school or general biology or both, mitochondria are double membraned. They have an inner and an outer membrane. More importantly, that inner membrane is ruffled to increase its surface area, and the innermost compartment on the inside of the inner membrane is called the matrix. So if you were to go the deepest you could into the mitochondrial organelle, you would be in the matrix. The space between the two membranes is called the intermembrane space. So if you penetrate the outer membrane, but you haven't yet gone through the inner membrane, you are in the intermembrane space. So most of us are used to seeing mitochondria in kind of that kidney bean drawing, but if you were to take that mitochondria and slash it down the center, if you were to just kind of take a knife and draw and pull it down the center. So if we were to cut a mitochondria lengthwise and we looked into one half, this is what we would see. We would see all this membrane in there, kind of creating this spaghetti, this hollow spaghetti of membrane, and only in the deepest part of that area would we be in the matrix of the mitochondria. So the mitochondrial membrane, the inner membrane especially, is going to be critically important as we continue going through this material and continue metabolizing glucose. The outer membrane of the mitochondria is quite strict, but the inner membrane is much more strict than that. Essentially nothing can cross the inner mitochondrial membrane without a channel. Nothing can. Whereas we know in cells we can get the diffusion of small nonpolar molecules that float through the cell membrane kind of like a ghost through a wall. All of the chemical reactions that occur in the citric acid cycle are occurring in the deepest part of the mitochondria, in that matrix, and so we'll be living in there for probably the next 2 lectures and then some. There is one single enzyme of the citric acid cycle that is membrane bound and occurs at the mitochondrial inner membrane, not within the matrix, and that plays a very important role in linking the citric acid cycle to the electron transport chain, but we'll keep that for later. Some more key features of the citric acid cycle. Essentially, the citric acid cycle is going to be a bunch of redox reactions. No surprise, because we know that the citric acid cycle exists to harvest electrons. As we reduce electron carriers with the electrons from glucose, we are oxidizing those glucose carbons. We are fully oxidizing those glucose carbons so that they become carbon dioxide. That's the carbon dioxide we exhale. The electrons, now reducing those electron carriers, are going to be shuttled to what I've been referring to as the electron transport chain, where they will be used, their energy will be used to drive more metabolic processes forward. Slowly, little by little, the high power energy in those electrons will be released and used to drive a process that will allow us to make ATP. So we're talking about oxidizing pyruvate. We're talking about stealing electrons from pyruvate, causing those carbons of pyruvate, the carbons that originally belonged to glucose, to become carbon dioxide, and also to allow acetyl to be made. So as this carbon dioxide is released, at least one carbon dioxide is released, we can link the acetyl to CoA and kind of pair acetyl up with that very uncomfortable partner, making it more likely to engage in the chemistry of the citric acid cycle. In the citric acid cycle itself, 2 more carbon dioxides will be removed from acetyl-CoA. This will be done through what is called decarboxylation reactions. Our electron carriers will be NAD most of the time. There'll be one case where FAD is reduced instead, and these electron carriers are what will bring the electrons to the electron transport chain. Of course, if we're saying that these carriers are reduced, that means they have gained electrons. They have gained electrons because the carbons of pyruvate, that is the carbons of glucose, have lost those electrons. Those electrons, once carried past the electron transport chain, are going to have their energy used for a process called oxidative phosphorylation, and that's where we make ATP. That phosphorylation in that phrase refers to phosphorylating ADP into ATP. But I want you to note, we're talking about losing a single carbon dioxide molecule on our way to making acetyl. So acetyl is a 2-carbon molecule, and pyruvate, of course, has 3 carbons. So when we convert pyruvate into acetyl, we lose a carbon dioxide. Furthermore, I tell you that within the citric acid cycle itself, 2 more carbon dioxides are lost from acetyl. So if we count them up, we lose 1 carbon from pyruvate when we make acetyl. We lose 2 more carbons from pyruvate when we oxidize that acetyl to steal its electrons. If you count them up, that's 3 carbons. That's the 3 carbons that made up pyruvate. So for every 1 molecule of glucose we consume, we make 2 pyruvates, and we lose carbon dioxide from those pyruvates as we make acetyl and process acetyl. So pyruvate, and therefore the glucose that was used to make it, is gone. All those carbons exhaled. So that means as we ate glucose, transported it into our cells, went through that 10-step process to convert it into pyruvate, we were never interested in the carbons of that glucose at all. How interesting. It was the electrons we were after. The electrons are the energy of glucose, and that's what we need. So everything we're going to think about and talk about now, we're going to stray away from kind of the tangible materialism of glucose and begin thinking in larger terms of energy. Energy manifest in the form of electrons. Everything I've just described, releasing all of those carbons as carbon dioxide and essentially completely decimating glucose in the truest sense of the word so that we get all of the electrons that we need, that all happens in the first 2/3 of the citric acid cycle. The last third of the cycle is dedicated to allowing it to be just that, a cycle. It's allowing the citric acid cycle to regenerate back to where it started so that it is a process that can repeat again and again. So this lecture chunk is meant to just kind of bring you up to speed on where we're heading and why it's important, and then we'll use the next 2 chunks to drive these messages home and start introducing you to the reactions of the citric acid cycle itself. Until then, thanks for watching.